Students, in previous class we were discussing regarding the various aspects of immunity with respect to human health and diseases. In today's topic we are going to discuss about a very important, a fatal disease for the human beings which is being spread from one to the another. That is highly infectious, a fatal disease for the human being is AIDS. Let's discuss about that AIDS now. See, the AIDS is nothing but it is regarded as acquired immunodeficiency syndrome. So why it is called as syndrome rather than a disease? So AIDS is rather than a disease, it is a syndrome. Why? Because when a patient infected with or when an human individual get infected with HIV, that is infection by that is human immunodeficiency virus, that is HIV, it causes a condition which weakens the immune system of such individual, and then that person become opportunistic for various diseases. So that he will be suffering from many diseases at a time. So only such condition is called as a syndrome, which is because of the deficiency of immunity, and that is caused by what is that? Infection with the HIV. So as the human is going to acquire this disease in his lifetime, so only it is called as acquired immunodeficiency syndrome. So AIDS means acquired immunodeficiency syndrome. Now, so it is acquired during the lifetime by getting exposure to the infective or the infected person, either through the blood transfusion or through the sexual contact. So only it is acquired syndrome, and it is not a congenital disease. It is not a congenital disease, but it can be transmitted from infected mother to the fetus through the placenta. So such a AIDS was first reported in which country? Means that is USA, United States of America, that is in 1981. And this AIDS was first reported or discovered among homosexuals in the USA. What do you mean by homosexuals? Means here the sexual activity between the male and male and male, or female with the female. Such individuals are called as homosexuals. Among the homosexuals, the first incidence of HIV infection or the AIDS was reported in USA. Next is, then this syndrome was widespread worldwide and killed more than 25 million people. Such an infectious disease this AIDS is. So how infectious this AIDS means, or the HIV infection means, it killed more than 25 million people all around the world. So such AIDS was detected or discovered in India in 1986, and the first case of AIDS was identified in Madras or the Chennai in India. That is during 1986. Remember the numerical that is here. The first incidence of AIDS was discovered in USA, that is 1981, but in India in 1986 in Madras, and now it is called as Chennai. Next, what is the pathogen for such AIDS disease? So the pathogen for the AIDS is, as I said earlier, HIV, that is human immunodeficiency virus. So because of the nature of the deficiency what it causes in the human being, so it was given with the name HIV by the International Committee for Virus Nomenclature. So such HIV belongs to a group of virus which is called as retrovirus. What do you mean by retrovirus? Means these viruses are capable of synthesizing DNA from the RNA. So only they are called as retrovirus. So such an HIV virus, they have an envelope which is made up of bilipid layer. The envelope of the virus, if you observe in this diagram, this is the structure of the HIV virus. So here what happens, in this diagram there is the presence of two lipid layer. So bilipid layer is present. Envelope of that HIV is a bilipid layer, which is derived from the host cell membrane. Means from where this bilipid layer is derived means it is derived from the host cell membrane. Means for example, when that HIV virus is infecting a human individual, from the cells of that human body only, this bilipid layer will be derived or synthesized. And also have projecting knob-like glycoprotein spikes. See here, along with the envelope, they are also having projecting knob-like glycoprotein spikes. These glycoprotein spikes are made up of glycoprotein 120. What is the protein present in that spikes means glycoprotein spikes? That is glycoprotein 120, that is GP120. And along with the pedicles. These pedicles are made up of protein called as GP41. So, these glycoprotein pedicles are virus coded glycoprotein. Means here bilipid layer is derived from the host cell membrane, but this glycoprotein spikes and glycoprotein pedicle, these are coded by the virus. So those are the virus coded proteins synthesized, and these spikes are having GP120 protein and their base, that is the pedicle, is having GP41 protein. So if you observe the structure of the HIV, this is what the structure of HIV is. See what it is having? Bilipid layer, and along with that one the glycoprotein spikes studded in that bilipid layer. See, if you observe here, see this envelope encloses the RNA. So the envelope of the virus, for example, this is the envelope of the virus which is having RNA, that is nothing but the two single stranded RNA filaments as their genome means genetic material. Along with that one, they are also having a reverse transcriptase enzyme. So two dot-like structures you can see in this diagram of HIV. So this indicates reverse transcriptase enzyme. Reverse transcriptase enzyme responsible for reverse transcription. Okay. Next, the HIV, such an HIV consists of a core with RNA and reverse transcription. See, for example, this is what the core of the virus. In this core, there is a presence of RNA strands along with the enzyme, reverse transcriptase enzyme. So this core is surrounded by the protein coats. The viral core is surrounded by protein coats. Two protein coats are present here. The inner protein coat is called as capsid, and it is made up of a protein called as P24. The inner viral protein coat is made up of protein called as P24. Similarly, okay, next, an outer layer of coat, protein coat, is made up of a protein that is called as P17. It is called as P17. And the outermost envelope is the bilipid layer, and this bilipid layer is studded with glycoproteins, that is nothing but GP120 spikes and a pedicle with GP41. This is what the structural representation of HIV. This diagram is very important for the PU examination. It will be asked for 2 marks in PU examination. Just make a note of this diagram and these points now. So, such HIV virus belongs to a group of virus called retrovirus, as I said earlier. What do you mean by this retrovirus? Means these viruses can synthesize their DNA by using the RNA filaments with the help of an enzyme called as a reverse transcriptase. As you already seen in the diagram, their genome contains RNA filaments with the enzyme reverse transcriptase. This reverse transcriptase helps in conversion or synthesis of DNA by using these RNA filaments. That is called as reverse transcription. So as they can carry out the reverse transcription, so they belongs to a group of virus called retroviruses. Then, what is the major cell get infected in our body with HIV infection? Or which cells the HIV infects majorly in our body? Means it is nothing but the helper T lymphocytes or TH cells. That is helper T cells or the helper T lymphocytes. Those are the major cells get infected with the HIV infection. That too, that is, these T lymphocytes will be having CD4 receptors on their surface, and HIV binds to that CD4 receptor sites. Name the receptor site for the HIV infection or the receptors on the TH cells into which the HIV binds, means that is CD4 receptor sites which are present on the T helper lymphocytes. So the attachment of the virus to that CD4 receptor, attachment of this virus to the CD4 receptor of which one? That is the T lymphocytes or T helper lymphocytes takes place with the help of GP120, that is glycoprotein 120, which are present on the, what is that one? Spikes of the glycoprotein. These glycoprotein spikes helps in the attachment of that virus to the CD4 receptor of the helper T lymphocytes during infection. Then, what is the mode of infection? What is the mode of infection or the transmission? How it get transmitted from infected individual to the healthy human being? So, the mode of transmission of the HIV includes, see, HIV can be transmitted, or transmission of the HIV can occur by the various modes, such as the first one is sexual contact with the multiple partners or unknown partner which are infected with HIV. Means sexual contact with the infected person can leads to the transmission of the HIV to the healthy person also. That is one way of transmission of the HIV. Similarly, by the transfusion of contaminated blood and blood products, transfusion of the blood and then the blood products contaminated with the HIV, means a person who is having the HIV infection, in his blood and as well as the blood products, the HIV will be present. Transfusion of such a blood and blood products leads to the transmission of disease to the receiver also, or the healthy individual. Next is by sharing the infected needles. By sharing infected needles. What is the meaning of that one? For example, if I am an intravenous drug user, means I used to take the drugs directly into my veins by needles, means if the same needle is used by you or the other person, if I am having the HIV, means that needle will also get infected with the HIV virus, and if the same needle used by you will transmit HIV to you also. In this way, in intravenous drug users, what happens? Through the infected needles, the transmission of the HIV, no need of any transmitters required, that is the needles or the blood or anything. As the fetus is growing in her womb, if that mother is infected with the HIV, means these HIV virus can cross the placenta and confer that disease to the fetus also, the developing fetus also. So, in that way, the child will also get the HIV if his mother or if its mother also get infected with the HIV. So these are the various modes of transmission. Then, okay, though means which group of human individuals are at high risk of getting the HIV infection? Which group of human individuals are at the high risk of getting the HIV infection? So the group of human individuals who are at the high risk of HIV infection includes, means for which human individual HIV may easily attack. It includes, one is those individuals who have multiple sexual partners. If the one or those humans which are having multiple sexual partners, okay, knowingly or unknowingly, they may be subjected for HIV infection. If any one of them is having HIV infection, means all of them will get the disease if such people come in contact with the infected person through sexually. Next is drug addicts. those who are addicted for the intravenous drugs, particularly intravenous drug users, by sharing the needles, okay, they will also be the victim of this HIV infection. Next is individuals who require repeated blood transfusions. Those individuals which undergo the repeated blood transfusions, that also leads to high risk of HIV infection. Okay, so these are the people who are at the high risk of HIV infection. Along with that one, last but not least, what is that one? Child born to the HIV infected mother will also be at the high risk of getting HIV infection. Then, by which means HIV do not get transmitted means, see, HIV do not merely get transmitted by touch or hug or physical contact, but it can be only transmitted through the body fluids, that is the blood and semen and blood products. By the transfusion of these body fluids from infected person to the healthy individual, there will be a transmission of the HIV. And what is the incubation period of this HIV? After gaining entry into the human body, when this HIV causes AIDS disease means, or the AIDS condition means, it takes few months to many years. Then what is the normal incubation period of this HIV means? It varies from few months to few years. That is, it may show its symptoms after 5 years of the infection or after 10 years of the infection. Sometimes even it may require 12 years also. So usually the average incubation period for the HIV is 5 to 10 years. So make a note of these points now.